Welcome to my channel Knowledge Seekers. In this lecture, you are going to learn the development of gonads, by which we mean the testes and ovaries. We are going to discuss the development of gonads in mammals, so in humans as well. They are mammals. But before going, and yes, we are not going to discuss any underlying mechanism. We are just going to go through the stages in this lecture. We are going to keep the things a bit simple. This lecture. So before moving on to this topic, I need you to know some important aspects of this. Well, the very first thing to remember is that in mammals, the sex determination, it has a couple of stages. The first stage is called primary sex determination, and the other one is called secondary sex determination. And before going into the definition of that, another important aspect that I need you to know is that unlike the other organ rudiments in mammalian embryo, the gonadal rudiment, they are what you can say they are bipotential, or what you can say a gonadal rudiment. It can either uh, be determined to form the testes, or it can be determined to form the ovary. And normal cases, what happens is that the lung rudiment is going to form the lung, and the heart rudiment is going to form the heart, not being exact. But in the case of gonads, the gonadal rudiment, it can form either of the two gonads, testes or ovaries, that are quite different in their tissue, what you can say, organization or tissue architecture, etc., and their function as well. So the primary sex determination is the determination of gonads, that whether that gonadal rudiment is going to get determined to be testes or the ovaries. This is called the primary sex determination. Then what happens is that when gonads have been determined, those gonads, they start producing hormones. For example, let's say case of ovaries, estrogen, etc. In case of testes, testosterone, etc. So these hormones, they are going to, what you can say now, cause the phenotype of the male or female to be obvious, or to, what you can say, be produced. And that, what you can say, effect of hormone on body, shaping the bodies at male or female, that is what is called the secondary sex determination. So first of all, the gonads, they are determined to be either of the two gonad types, and then the gonad types that is the gonad type that determined, it is going to, what you can say, secrete its hormones, which is going to lead to the secondary sex determination. Now let's come to the topic. But first, here we have, what you can say, the gonadal rudiment, or what you can say, a bipotential or sexually indifferent gonad. Now this gonad, it can end up being either as testes, these couple of gonads, or as ovaries. If that genotype of that, what you can say, embryo, it is XY, then it is going to get developed into the testes. However, if it is XX, then it is going to get developed into the ovaries. Another important thing is that in this sexually indifferent, or simply the indefinite gonads, or the bipotential gonads, or the gonadal rudiment, you can see that both the Wolffian duct and the Mullerian duct, both are present. But in, what you can say, males, only the Wolffian duct is present and the Mullerian duct is degenerated. And in case of females, the Wolffian duct is degenerated. However, the, what you can say, Mullerian duct, it is present. And in, we're going to get into the detail of that, but I just need you to know these couple of things, that what is going to change into what, and what is going to stay and what is going to go. So in testes, the germ cell location, it is inside testis cord, in the medulla of testes, and in ovaries, this germ cell location, it is inside the follicles of ovarian cortex. In testes, or what you can say, in males, the Wolffian duct remains, and in females, the Mullerian duct remains. The other duct in males, Mullerian duct is going to get degenerated, and in females, the Wolffian duct is going to get degenerated. Now, this Wolffian duct in males, it is going to get differentiated into vas deferens, epididymis, and seminal vesicle. And in females, this Mullerian duct is going to get differentiated into oviduct, uterus, cervix, upper portion of vagina. The urogenital sinus is going to form the prostate gland in males and Skene's gland in females. And what you can say, the labioscrotal fold, it is going to end up as scrotum in males and labia majora in females. And the genital tubercle, it is going to end up as penis males and clitoris in females. So now let's come to our topic. So we know that in humans, two gonadal rudiments appear during the fourth week and they remain sexually indifferent until the week 7 of pregnancy. So, until the week 7 of pregnancy, what you can say is that uh, there are no gonad, there are just gonadal rudiments or gonadal precursors. Now, these gonadal precursors, they are paired portions of mesoderm adjacent to the developing kidney. If you remember what you can, the stages of kidney development, another lecture present on my channel. In that, we kind of realized that gonads are quite near the developing kidney. So these gonadal precursors that appear during the week 4 of pregnancy and that remain sexually indefinite until week 7, they are paired regions of mesoderm that is adjacent to the developing kidney. And the germ cells or the precursors of either sperms or eggs, they migrate into gonads during the week 6 and are surrounded by the mesodermal cells. Now take a look at this diagram. This is a week 4 embryo and we are looking at the indefinite gonads. So here you can see that this is a mesonephric ridge and here we have the excretory mesonephric tubule. We know that mesonephros is the second transient kidney or the second stage of kidney development. So the important thing here to notice is this the presence of Wolffian duct as well as what you can say presence of the Mullerian duct as the week 6 comes. So both of the ducts are present in these bipotential or indifferent gonads. And then we talked about the migration of the germ cells, as we can see here, the somatic and germ cells, they are getting migrated into the gonads, into the indifferent gonad. And then what you can say, the mesoderm is surrounding these. If the fetus has a genotype XY, then the mesodermal cells, they continue to proliferate through the week 8. And when a subset of these cells initiate their differentiation into Sertoli cells. So if it is going to be a male, and so the indefinite gonad is going to get developed into the testes, then the mesodermal cells, they are continuing to divide through the 8 week or what you can say till the week 8, till the end of week 8, and a subset of or what you can say a small group of this mesodermal cell, it is going to get differentiated into Sertoli cells. And the developing Sertoli cells, they secrete anti-Mullerian hormone. And this anti-Mullerian hormone, it is going to get block the development of the female ducts. And the same Sertoli epithelial cells, they will also form the seminiferous tubules that will support development of sperm throughout the lifetime of the male mammal. We know that we have just taken a look at it in males. This Mullerian duct, it is going to get degenerated, and how it is going to get degenerated? Well, in simple, it is going to be because of the effect of these developing Sertoli cells that are secreting anti-Mullerian hormone, and this anti-Mullerian hormone is involved in the degeneration of Mullerian duct in males of mammals. And then during the week 8 of pregnancy in human females, in humans, human embryo, the developing Sertoli cells, they surround the incoming germ cells and they organize themselves into the testis cords. And these cords, they form loops in the center region of developing testes and are connected to a network of thin canals that are called the reed testis, and they are located near the developing kidney duct. Let's take a look at that. So here you can see that the week 8, now it has been determined that whether it is going to be a testis, that in different gonad it is going to be a testis, or it is going to be an ovary. So in case the genotype is XY and the gonadal rudiment is going to get determined and then differentiated into testes, then we can see that this Wolffian duct it is going to develop into the vas deferens. This Mullerian duct is going to get degenerated in a while, and we can see here the degenerated mesonephric tubule, because we know that this second transient state of kidney is going to get replaced by the final kidney or the permanent kidney that we call the metanephric kidney. And here what we talked about the reed testis cords, you can see here, and here we have the germ cells, and here we have the testis cords, and here we have the tunica albuginea. These are all terms that I'm not going to describe in detail here. Just the terms that are the topic. Already studied the testis cords and the reed testis cords. Now, when the germ cells they enter the male gonad, they develop into the testis cords inside the organ, and later in the development at puberty in humans, short after birth in mice, because they procreate much faster. The testis cord it matures to form the seminiferous tubule. So this testis cord it is going to develop into what you can say the seminiferous tubules, and the germ cells they migrate to the periphery of these tubules where they establish the spermatogonial sperm cell population that produces sperm throughout the lifetime of male. So in males, sperms are produced throughout the lifetime. Any important thing, any important difference from that of females. Now, in the mature seminiferous tubule, the sperms are transported from inside of testes through the reed testis, which joins the afferent ducts. And the afferent ducts, they are remodeled tubules of the developing kidney. Now, during the male development, the Wolffian duct, it differentiates to become the epididymis, and it is present adjacent to testes. And the vas deferens, the tube through which sperms they pass into urethra and out of the body. And both sperms and urine they use urethra to exit the body in human body and in the body of all the mammals as we know. So here we had the 8th week embryo of a male, and then in the 16th week we can see that this Mullerian duct it is going to get degenerated. This Mullerian duct, green one, it is going to get degenerated. Sorry, yes, yellow one, it is going to get degenerated. However, this Wolffian duct it is going to get differentiated into the vas deferens, and the afferent duct here we can see the vas deferens or the tubules that are going to take these sperms outside of the body via urethra. And here we have the reed testis cords. You can see here we have the tuna albuginea and we have the spermatogonium. And then we have the testis cord. So this 8th week embryo it is going to gradually develop into this 16th week stage that will look like this. And this testis cord, these are what are going to form the seminiferous tubules in the later stages of development. As you can see, it is just the 16th week. Now, the other group of mesoderm cells, those that didn't form the Sertoli epithelium, they differentiated into what you can say a mesenchymal cell type, which we call the Leydig cells. And these Leydig cells, they have a function that they secrete testosterone. Thus, the fully developed testes, it will have an epithelial tube. It will have epithelial tubes made of Sertoli cells that enclose the germ cells as well as the mesenchymal cell population, the Leydig cells that secrete testosterone. And each incipient testis is surrounded by a thick extracellular matrix, the tunica albuginea, which helps protect it. So this tunica albuginea, it is the EM or extracellular matrix protecting the testicle. Now let's say that instead of XY, the fetus is XX or the female genotype. Then the sex cord in the center of developing gonad they degenerate, and this leaves the sex cord at the surface or the cortex of gonad. And each germ cell it gets developed, it get enveloped by a separate cluster of sex cord epithelial cells. Take a look at it. As you can see in the center here, the cords they have degenerated the sex cords, what you can say the ovarian cord. These were the testis cords. They have degenerated, and here we can see only the cords at the surface of the gonad or at the cortex of gonad. They are left. In the center, they have degenerated, as said earlier. And each of the germ cell is going to get enveloped by a separate cluster of the sex cord epithelial cells, and the germ cell they will become ova in this case of females. In case of males, the germ cells they lead up to what you can say spermatogonium, I think, yes. And the surrounding cortical epithelial cells they will differentiate into the granulosa cells, and the remaining mesenchyme cells of the developing ovary they differentiate into the thecal cells. So if you what you can say, let's take a look at female productive tract or from the textbook of medical physiology, use that as a reference. So here I have the textbook of medical physiology 14th edition, uh, open in order to take you in order to make you take a look at the granulosa cells and the thecal cells. So here we can see the multiple layers of cell. This internal layer of cell, it is the granulosa cell. We are seeing the various stages of follicular growth in female reproductive cycle. So these are granulosa cells, and this the external layer of cells, it is what we call the theca. And so these cells, this external layer, it is going to be the thecal cells. So this is what I was trying to show you, the thecal cells and the granulosa cell. Now, together the thecal and granulosa cell, they form follicles that envelop the germ cell and secrete steroid hormones such as estrogens and when pregnant, progesterone. And each follicle, it contains a germ cell, an oogonium or egg precursor, which will enter meiosis at this time. Here you can see we have these granulosa cells and thecal cells surrounding are this uh, oogonium. As you can see on your developmental biology textbook, during the 20th week of pregnancy, the female fetus, its gonads look like this. Here we have the ovarian cortex, and this is the surface epithelium of the ovary. And here we have the oogonium inside this follicle, which is what you can say the layers of uh, granulosa cells and the thecal cells. Here we can see that the Wolffian duct it is going to get degenerated, as we can see here. However, the Mullerian duct it is going to be present, and it is going to get developed into a number of uh, parts of female reproductive system that we studied a while ago. Now let's get to a very brief mechanism. Well, there is a reciprocal relationship between the germ cells and the somatic cells of the gonads. We know that a couple of types of cells are present there: the sperm cells that are making up the gonads, and the somatic cells that are making those gonads uh, structurally, and the germ cells that are going to what you can say end up being gametes. Now the germ cells they are originally bipotential, and they can become either sperms or eggs. And once in male or female sex cords, they are instructed to be either what you can say begin meiosis and become eggs, or remain meiotically dormant and become spermatogonia. And in XX gonads, the germ cell they are essential for maintenance of ovarian follicle, and without germ cell, the follicle degenerates into cord-like structures and express male-specific markers. So, in the XX gonad, this germ cell, their presence is pertinent. And in XY gonads, the germ cell, they help support the differentiation of Sertoli cells, although testis cord will form even without the germ cell, albeit a bit later. So, in female, what you can say that development is entirely dependent on the presence of germ cells. However, in the case of males, they have just a supporting role and not what you can say a vital role. And when an ovary is being formed, the Mullerian duct it remains intact because there is no anti-Mullerian hormone or AMH to destroy it, as in the case of the development of testes. And this Mullerian duct is going to differentiate into oviducts, uterus, cervix, and upper vagina. And in absence of adequate testosterone, the Wolffian duct degenerates. We know that in males, the Wolffian duct it's are going to get degenerated normally, but if testosterone is not present, then this Wolffian duct is going to get degenerated, as we talked about it in the very beginning of lecture, that the secondary sex determination is going to be because of the hormones that these uh, gonads that have determined to be either testes or ovaries are going to produce. So if the testes they do not produce testosterone, the ladies they do not produce testosterone or they are not produced themselves, then that Wolffian duct is going to get degenerated, and a degenerated Wolffian duct means that we are not going to have a couple of crucial parts in the male reproductive system, such as the vas deferens. So these were the stages of the development of gonads in mammals. We took a look at what is the indifferent, sexually indifferent gonad or the gonadal rudiment, or what you can say the bipotential gonad, and then we briefly uh, try to understand how it is going to get determined to be either the testis or ovaries formed by step by step. So I hope that you have found this lecture to be helpful, and if it is so, then please do like this video and do not forget to subscribe and turn the notifications on. Maybe later in a video we will go after the mechanism that is underlying all this primary sex determination and secondary sex determination in mammals. Thank you so much for watching.